When Dr. Anthony Fauci spoke recently at a White House briefing about the need for COVID-19 booster shots, buried in his slideshow of charts and data points was a little-noticed scientific paper that offers evidence for a reliable way to predict how much protection a COVID-19 vaccine offers. The study appeared on a preprint server earlier this month without much fanfare, but many interested in the future of COVID-19 vaccines had been eagerly awaiting the results. The researchers were looking for markers in vaccinated patients' blood that would indicate protection against COVID-19, what's known as correlates of immunity. What the team of scientists found were neutralizing antibodies, proteins made by the immune system that are known to disarm the coronavirus. As Fauci explained, the paper showed that higher levels of these antibodies are associated with higher levels of vaccine efficacy. The findings suggest that giving people a booster vaccine, which has been shown to raise antibody levels, would go a long way toward protecting them against the coronavirus, including some of the newer and more dangerous variants. While more studies are needed to confirm the findings, discovering that these markers correlate with immune protection has implications for future COVID-19 vaccine research. It means that researchers now can measure whether a new COVID-19 vaccine might work, without necessarily having to repeat large-scale efficacy studies. That could be used as the basis for authorization and approval of vaccine candidates without needing to do these trials with 40,000 people that take a long time and a lot of expense to complete, says Peter Gilbert, a biostatistician with the Fred Hutchinson Cancer Research Center, the lead author on the new study. To understand if there is a protective level of neutralizing antibodies, a team of researchers from academic institutions, industry and the government did new research on the blood of people who participated in the large trial of the Moderna vaccine. That older trial, involving 30,000 volunteers, was the basis for the Food and Drug Administration granting Moderna emergency use authorization for its COVID-19 vaccine. This new research found 46 people in the Moderna study who had been vaccinated, but subsequently got sick with COVID and compared their levels of neutralizing antibodies with the levels found in a sample of 1,000 people who were vaccinated during the trial and never got sick. The antibody levels were always lower in the vaccinated people who became a COVID case compared to people who remained free of COVID, Gilbert says. The results show that antibody levels can be predictive of immunity, which should help develop and test new vaccines at a much faster pace. It's even possible COVID-19 vaccine makers may not need to conduct trials with huge numbers of people to see how many get sick after getting vaccinated.